if you're good with geography, you might see a sort of pattern here. And if you're good at knowing these two-letter labels, too, you might start noticing a pattern here. We've got Spain and Portugal, France, Italy, CH is Switzerland, BE is Belgium, GB is Great Britain, Scotland, Ireland. Is anybody seeing a pattern? Yeah. Yeah. Okay. Too good. All right. So that's super. And yeah, so there's the map right next to it. And so these genome-wide patterns of variation, they match with the geography of Europe. Now there's a couple, that's the broad picture, but there are lots of little details. So you've got Great Britain here. This is here's an individual from Great Britain, but they look like they're from southern Italy. Okay, this is probably somebody who was living in London, sampled from the UK, and has Italian ancestry. Okay.
So let me ask you a practical question.
Yeah.
Let's say we took some of my DNA or some of the DNA from people in the room, students in the class. So let's just say it's me, and we ran through this chip and did this analysis. Then theoretically one would be able to pinpoint on this map that perhaps my ancestors came from Hungary or Poland or some part in Eastern Europe. Would that be the...
That's an application.
That would be an application.
Yeah. And I'll get into that.
And so what would happen if someone with Asian ancestry was... Where would they fall in here, or they wouldn't? Someone distantly related to what your group, how would that lay down?
So on just a European-only analysis, they'll fall out as an outlier. It'll just be off. But you can rerun it with, we have East Asian samples, and then they would show up in East Asia. Okay. So a couple of points I want to make. So you have, it's not this perfect map. There are lots of little individuals that are either mistakes in the way the tubes were labeled during all of the lab handling, or they represent individuals who are recent migrants, where we didn't have their grandparental information, and so we don't know that their ancestry is actually from somewhere else. So that's kind of reflected in the graph. Then there are a couple places where it doesn't match perfectly. So does anybody know what SK stands for? Slovakia, okay. And so Slovakia, right next to Czech Republic. You would think this point should be sort of up here, near the Czech Republic, Hungary, but it's actually way down here, okay. And is that because there's some deep connection between Slovakia and Italy? It's probably not, because we only had one individual from Slovakia, and they were probably actually of Italian ancestry, and so that point gets moved down here. The median of one individual is just that one individual's position. Okay, so otherwise the rest of the positions are roughly where you'd expect them to be. These were sort of fascinating. These 5 Italian individuals that seem to be floating out here somewhere in the Mediterranean, and we weren't sure, what to say about it, and we put a sort of speculative line about, well, they might be, since we don't have very precise geographic information, all we know is that somebody's from Italy. They could be Sicilian or Sardinian. And I actually was able to merge the HGDP data set, which has a number of European populations, with this data set and rerun the analysis. And the HGDP has Sardinians. And so when you rerun it, you get this big clump of Sardinian individuals right where these 5 are sitting. So they probably are just 5 Sardinians that are in the sample, and all we know about them is they're Italian. So that's kind of cool. It's nice to kind of wrap that mystery up. Rose?
How do you determine what data compression you're using in order to get the PC, like PC1 and PC2, in order to actually have it be that, clear visual map?
Okay, so you have a whole choice of methods that you could use. And PCA is attractive in that for genetic data analyzed in the way that we were analyzing it, converting it to those 0, 1, 2 scores, it is well known that if you have clusters, it divides, it will, if you simulate from like discrete populations, you will get discrete clusters in PCA space. It's also known that if you have clinal model, that you will get this sort of map-producing property. So it's sort of interesting to do this and see, do I get maps or do I get clusters? And we get something that's more of a map. That's why we chose to use this method over alternative compression algorithms, yeah. But there wasn't like a vast amount of tweaking to get this result. This was sort of running it, and it's like, oh my gosh, you know. There's a couple things that you need to know that, like, to make it really hardly makes a difference, but PCA will be sensitive to if there are different sample sizes from different regions. So if I had like a very large number of Swiss individuals, it would start to just focus on all the Swiss variation and divide that up. Because it's trying to, in a data compression kind of way, it's trying to get the most bang for the buck. If most of the data is Swiss, then I'm going to try and explain the Swiss data as well as possible and ignore the rest of it. And so one of the steps is to, because there was two of the sampling localities were in Switzerland and the other one was in London, the major ones, we had lots of Great Britain individuals, lots of Swiss individuals. So I actually throw out some of those before running this. Yeah.
So John, what kind of time framework are we looking at here?
In terms of history?
Yeah, in terms of history.
Yeah, we don't really know.
Don't really know. I mean, obviously these populations had to be separated quite a bit in order to get this divergence here.
Mm-hmm.
But are we talking about tens of thousands of years? And I mean,
yeah, it's a great question, and right now we don't have the methods to take SNP data and do that sort of estimation. So we can do it for sequences because we can look at the number of mutations in an area and count up the number of mutations, and it gives us a sense of, if we know the mutation rate, we can work backwards to get the amount of time that's passed between those two sequences separating each other. That's what went on with the mitochondrial DNA analysis. But these SNP chips, you're only looking at sites that you know are variable. So it's not random whether something is variable or not. You've you've built this chip to just look at variable sites. So you have this problem that you can't kind of calibrate it to a time. And there are some approaches. This is kind of ongoing methods development, some of the computational challenges. We'd obviously like to know. But the levels of differentiation, even though you get this very nice picture, are extremely low using some population genetic statistics. So it suggests that times are very, very, very recent, but I don't have an exact date.
But
yeah.
the data, and I think all of us would really understand, and if you think about it, the data really points to historical isolation of populations, even though from our point of view, these are pretty close. You know, way back when, there was no way to go from one place to another, and one would speculate that some of these people who lived in these areas probably stayed for maybe thousands of years in the same area with respect to their families and their ancestry and things like that, with relatively little migration in effect, because if there was a lot of migration, you'd never see these kinds of patterns. There'd be a lot more mixture and stuff. And so it said that there's, to me, these data say that there were like isolated local populations way back when doing their own thing and staying within a rather narrow geographic history. And I would imagine, again, for those of you that are doing this, that's reflected in linguistics, and many of things probably dovetail with linguistic studies and those kinds of things as well.
Yeah. Getting at linguistics, an actual side story that's pretty interesting is Hungarians speak a Finno-Ugric language, which is, it's actually different from all the other languages that are spoken in Europe. It's not even, there's even a larger family of languages that European languages fall into called Indo-European. And Hungarian's different from that altogether. And so you might think that if genetics and linguistics were perfectly tied together, that Hungarians would sort of pop out outside of the map, but they appear where they should be. And I've had sort of historical demographers listen to the talk, and they've said, oh, but that makes perfect sense because it's known that Hungary was overtaken by an elite that changed the language, but all the people just were the same people after they started speaking Hungarian as before. So that kind of speaks to a very localized population.
We had a
Tiger.
So, how you were saying how you are like from Uruguay and Italy, and a lot of people in the United States, I feel like especially are like this, where you wouldn't see a map at all that looked like this. If you sampled people from all over the United States, you wouldn't see like the different states pop out. And with like the transportation advances like Julie was talking about, do you think if you tried to do something like this, even 50 years from now, you wouldn't get divisions?
In Europe.
Yeah.
Yeah.
Or what if you tried to do this in the United States like today?
Yeah, so if you did this in the United States, what you would get is if you say you use the HGDP, you had the Human Genome Diversity Panel as a kind of backbone, and then you took everybody in this room, then those of us with East Asian ancestry would go with the East Asians, right? And those of us that are admixtures, recent admixtures, we'd go in between the locations where our ancestry is from. And so that you can sort of see the footprints of our recent ancestors moving in this kind of plot. But it's not that you would see a little map of Florida and California and, yeah.
So like if you repeated this 2 generations from now, do you think it would be sort of a multicolored?
Yeah, it's going to get fuzzier and fuzzier, absolutely. Yeah. You'd see more of these kinds of events, right, where...
Has any of this sort of data or analysis been able to elucidate anything about the mystery of the history of the Basque people in Spain?
I guess I'm not ready to kind of say in detail what an answer is, but when you merge the HGDP, which has Basque samples, they kind of appear, right here. It's not that they are. So there are stories about the Basque that they represent a Paleolithic population that, and by that I mean prior to the invention of agriculture, there were groups in Europe that were living there for a very long time, and the thought is that you had an expansion of agriculturalists from the Near East that represent most of the ancestry of Europeans today. Even that's a debated hypothesis. But anyway, there's this idea that Basques represent a very ancient European lineage, and that the rest of Europe is somehow derived from a much more recent group of populations. And at least from that kind of result of the Basque appearing here, it's showing that they're not that strikingly different from their geographic neighbors. But that doesn't necessarily say what the Basque population history is. It just says that's what Basque individuals living today look like, right? It could be that the Basque population was quite separated and then started having gene flow with its neighbors, and so that today they look quite similar. But they actually might have been separated for a long while in the past. And I think that's why I don't really want to make a claim, because I think you need to look carefully for telling apart those 2 different stories. Yeah. Okay. But that's a great question. Okay, so you can kind of crank this up, and if you look, I said we had lots of Swiss individuals, so you can look within Switzerland. Oh, and even for the Swiss individuals, we also had what language they spoke. So you can look and you see that French-speaking Swiss looks slightly more French, German-speaking Swiss looks slightly more German, Italian-speaking Swiss look more Italian, so that you've got these subtle patterns of differentiation even within a very small geographic area. And then this is another paper that I wasn't involved with, but it's actually involved other researchers at UCLA. This is looking at Finnish samples, and these are different geographic groups within Finland and the PCA plot. And you see another kind of map-like result where the different regions are colored here. You can see how in the plot they match. There's sort of the geographic relationship is again recapitulated in this plot. This is multiple dimensional scaling, but it's very similar in principle to PCA and has the same behavior. So it's really quite striking how much resolution there is with this SNP chip data. Okay, and that might give you the mistaken impression that there are very different gene frequencies between different populations. And I'd mentioned that I was going to show you what it looks like at any given SNP, how different are the values. So here is, for 25 random SNPs, I've plotted the allele frequency on the y-axis for 6 regions of Europe. I grouped the populations to get the sample sizes to be larger, so I could get a good estimate of the allele, a better estimate of the allele frequencies, and then plot those allele frequency estimates across these 6 regions. And so let's take a random one. Okay, this green one. How much does it vary across Europe? It's like not even a percent, just a couple percent. What about this purple one? It's just a couple percent. What about this blue one? Okay, well, there's a little bit of a bigger dip there. But overall, these things are varying with just a couple of percent frequency differences across Europe. And this kind of points towards 2 things. One is that for any, it's really that the PCA method is pooling together all these very weak signatures of difference to kind of pull out that where each individual comes from. Okay. One analogy is that even though there might be, it's almost like where your ancestry is from biases a coin that you flip just very slightly. Okay. So that if I'm from Spain versus Italy, I might have a coin that's biased slightly towards heads. And if I just flip the coin once and I see that it's heads, I can't tell, or you can't tell whether I'm from Spain or Italy, right? But if I flipped it 500,000 times, you'd be able to see that it's shifting just away from 250,000, 250,000 in a way that's statistically significant that I'm from Italy or Spain, okay? So this is a pretty deep point because what it means too, that in terms of my biology, any one particular locus, right? If you wanted to guess and say, oh, he's from Spain, he's got this mutation, you're probably gonna, at locus X, right? You're probably gonna do a really crappy job because the biases are the differences are so small, okay? Now that's not true everywhere. That's what's really complicated about this picture. Most of the genome, that's true, okay? But there's some locations, a very few, that small percent where there are these stronger differences. But this is 25 random SNPs, most of the genome, and most of where the signal's coming from, it looks like this. This is, population geneticists use this number. It varies between 0 and 1. 0 means there's no difference between the populations. 1 means there's the maximum difference possible. For these European groups, it's 0.004, okay? For humans in general, it's 0.1, okay, across the globe.
John, let me weigh in for a second, if I may. Are you really saying that if you look across the genome, the allelic differences are not that great?
Right.
But there are places where you'd have large allelic differences, but that's the very small.
Right.
And those are in the things that we talked about when I talked about race and ancestry in human population, that most of the allelic frequencies, most of the alleles are there in different populations, and most of the allelic frequencies are the same. So the point was, with respect to discussion yesterday, when you would classify someone on the basis of skin color, those would be non-representative loci, because those allelic frequencies that represent color would be very high in one population compared to another. But if you look at the vast majority of genes across the genome in those 2 individual groups, they'd be the same. And that's where this race argument comes in.
Right, right.
Does everyone see that point? Okay, yeah. That's very important.
Yeah, and we're going to revisit that if I don't drag on too long. Let me know. Okay. Right, oh yeah, just a bit of trivia. I should kind of keep moving instead of side tracks, but lactose intolerance is the biggest thing that varies in Europe. So if there's like the gene that's responsible for how we digest milk is in much higher frequency in Northern Europe versus Southern Europe. Some people think that's because those cultures had dairy earlier than cultures in Southern Europe. And so there was a stronger selective pressure to be able to digest milk as an adult, and that drove all that differentiation. But anyway, so, okay, so just to summarize this about clines versus clusters, at a global scale, there's support for clusters and clines can be observed, and there's kind of this unresolved issue of, do geographic barriers lead to global scale cluster results, or is it uneven sampling? And the general feeling, yeah, and that we kind of came up with, we actually showed on the board, is that there might be some impact of geographic barriers, but if so, it's quite small. Okay, and then also we sort of showed how with large numbers of SNP markers, you can see patterns of differentiation even within continents, and those patterns at a continental scale are very clinal in that they look like, at least within Europe, very much like a geographic map. And we, again, are highlighting still that the variation is really between, or within groups rather than between groups. And this is all going to get pretty interesting because that's with 500,000 SNPs and even reduced down to 197 for technical reasons. But once we have whole genomes, we're going to have even more information and might be able to even look at even finer scale levels of relatedness. Okay, so an application of all this is for ancestry inference and personalized genomics, and you have these companies like 23andMe and DecodeMe and others that are allowing you to send in your DNA and get some answers about your ancestry. So here's our clicker exam again. I think you guys are just going to blow these out of the water like you did on the last one. So my mitochondrial DNA test comes back and says I have a Native American mitochondrial haplotype. Does this mean A, I is likely to be 100% Native American. My mother's side is likely to be 100% Native American. My mother's was likely Native American. D, not enough information to tell. Just want to click away real quick. It's probably too easy for you guys. Oh, that was the last one. Thanks. Oh, okay. Okay, click away. That's a hard one. Okay, we're good? Okay, stop. Display. Great, you guys are brilliant. All right. These UCLA undergrads, I have to get used to them. You know, at Berkeley, they're all just. Okay, so. All right, so great. So you guys got it, and you know this is something you guys are, you know, clearly college educated and ready to take on the world. But you know, you do have these for quite a while. People are getting these, you know, it's you know, like my uncle who's like, oh. I really want to get my Y chromosome type so I can find out whether I'm 100% German. It's like, no, you'll find out if, you know, if your father's father's father was, you know, German, say, right? And the point being here, like if you look at this pedigree and we imagine some individual, and the mitochondrial DNA is just going to tell us about this lineage. These are say male, female. This is the maternal line here, right? Paternal line. The mitochondrial DNA will just tell you about this particular ancestor. And so it's missing, just going back 3 generations, it's missing 7 of the 8 ancestors you have at that particular generation. Right? So you see a lot of people putting a lot of faith in their mitochondrial DNA results. You know, which daughter of Eve am I? There was actually a company that would tell you which of the 7 daughters of Eve you are. And kind of they were, you know, these people are defining their identities by their mitochondrial DNA haplotype when it just represents, you know, going 3 generations back, 1/8 of your whole ancestry, okay? And on the father's side, and for Y chromosome haplotypes, it's likewise just a part of your ancestry. So if I were to look at this person's mitochondrial DNA, we'd get that they're orange, but we'd miss that they have all of these other ancestries. And likewise for the Y chromosome. So that's something important to keep in mind. Okay, now we're going to think a little bit about admixed individuals and the pattern that it leaves in gene frequency. So picture we have an individual who has their ancestry at this time point comes from these different locations, okay, that we can label. We can say this person's from population blue, from population red, green, purple, and so on. And then they pass down chromosomes. And then those chromosomes recombine and get passed down. Okay. And this side's simple. They're all from the same ancestry. But what you see here is that you have this patchwork of ancestry on a particular chromosome. And using these high-density SNP genotypes, we can actually reconstruct, or attempt to reconstruct, these, this patchwork of ancestry. And that's the idea of chromosome painting that I'll show you in a second. But there's even an important point here still about what do I mean by ancestry. Because say I could reconstruct this, which is kind of all the possible ancestry information I could get from this person's DNA, you'll notice something that I would be able to recover that this individual has some ancestry from the red population, blue population, green population, and orange population. But what's missing? Purple, right? So there's some chance that I have ancestors of a particular ancestry that just by chance it doesn't make it to my genome, right? Which kind of raises questions about, again, if you're really trying to understand who were your ancestors just based on genetics, you're going to miss some of them, right? It's a very finite technology. So, right, if we have mixed ancestry, our chromosomes are going to be a patchwork of this DNA with different ancestries. By chance, we're going to lose some of the ancestors. And it's relative to time, right? So I'm sort of thinking about ancestry going back 3 generations. But each of these individuals has an ancestry that goes back, and their ancestors come from different populations. So, and if you go far enough back, everybody's ancestors come from Africa. So what does it mean to say, you know, what is my ancestry? At a deep time scale, you would say that your ancestry is all from Africa. At a very recent time scale, you'd say your ancestry is from California, I guess. And it's really that I think what people are curious about with their ancestry is kind of that intermediate time scale of where were each of my ancestors far enough back in time that they were kind of in one location, and their ancestors have been in that one location for a very long time. Does that make sense? You know, I want to sort of, the idea, I think people want to sort of trace their ancestry back to some village where their family happened to be living for a very long time, right? At each of the possible lines going back. Okay, so ancestry painting is this idea then of using the patterns of SNP variation to try to draw onto a chromosome those particular tracks of what part of your genome comes from one ancestry versus another. And right now, this is mainly done for individuals who are admixtures at kind of a continental scale. So many African American individuals or Latino individuals are going to have ancestry from different continental groups. So Latino individuals usually have ancestry from Native Americans and Europeans. African Americans tend to have ancestry from Africa and Europe and sometimes Native American. And so companies like 23andMe allow individuals to, you know, submit their DNA and get these kind of profilings, this chromosome painting, and this is an example of what you get. So what you can see here is like a whole section of chromosome 6 for this individual. Both sets of this individual's chromosome 6 appear to be of African descent, but then this part appears to be, they were heterozygous for a European chromosome and an African chromosome, and so on. And these orange parts reflect what they're saying is Asian ancestry. And this is really, yeah.
So would you call this person admixed or not? I mean, I guess that's just my confusion with what exactly admixture is, because, I mean, this person's classified as African American, but they have, you know, all these different other sort of ancestries present. So would this individual be admixed, or if they had a child with a European prominent person, then would their child be admixed?
No, so this person would be considered admixed, yeah, in that within their recent ancestry, they have ancestry from two different, very different populations.
So we're all admixed then? I mean, like, how many people have, like, all blue?
Yeah, so if you took, no, that's, I mean, but it's at what time scale and what signature the genetics is sensitive to. So, right, so if for this particular analysis, if you did it with a European individual, like one of them from my PopRes study, probably most of it would be blue, yeah. And it's that, African Americans have, it depends on, you know, on the individual, and that's part of why a lot of African Americans are interested in their ancestry, is to understand what portion have they inherited from Africa versus Europe versus Native American, and even of the African component, where in Africa is it from? So a lot of people like to know what part of West Africa are they from, and there's a.
So is that why for like the HAP map, when they picked the different groups, they didn't pick African Americans, they picked like Yoruban because of this sort of admixture with African Americans?
That's right. You get a very different picture of, they're very unique populations. Yeah, yeah. And this kind of analysis is very sort of cutting edge in the sense that like it's still being developed, and it's interesting that it's already kind of making its way onto these websites, because it's quite sensitive to issues of how you define even the reference population. So here you're trying to, you know, force it to be, okay, explain that part of the chromosome as either European, Asian, or African. And so kind of force each bit of the chromosome into those boxes. But what's odd here is that a lot of African Americans have some minor component of Native American ancestry because of histories of Native Americans and African Americans mixing in certain regions in the South. But here, there is no Native American as a reference population. And so a lot of places where the ancestry is probably Native American, because Native Americans have ancestry that's closer to Asian than either Africa or Europe, then where it's saying Asian, it probably means Native American. But even then, it's probably not getting it perfectly right. Okay? And so there's an issue with the reference populations that are being used here that can really confuse things, right? You have somebody looking themselves up on 23andMe and saying, wow, I'm 7% Asian, I had no idea, when it could be that they're actually Native American, have some Native American ancestry. So it's really complicated. I'm only trying to raise the complexity of the issues here, because I think that's the bigger part of the course, is to understand the complexities of biology and society and how these things can lead to a lot of confusion. And then in particular, you know, again, this is the result of statistical inference. These are all guesses, as you know, statistical guesses, best guesses. But with any statistical guess, you always formally think of a confidence interval, right? Some measure of your certainty in the estimate. And that's not being represented in this at all. So there are probably some places in the genome where you can get it quite well, you know, get the answer and be really sure that that part of the genome is of a particular descent, okay? But other parts where you're going to be less sure, and that's not being communicated to a consumer at all. There's not even a plus or minus 5 or 10% on any of these numbers, right? So I think there are a lot of interesting issues about how the ancestry inference is being used. But it's funny to be critiquing it, and then I'm going to show you an example of using it in my own analyses. I was interested in sort of showing from this European data, how far could you push this, okay? In part to raise the prospect that you might be able to do quite well, and that should make us all be thinking about ancestry inference a little bit more and trying to think more carefully about it. But the idea is that given we saw that map-like pattern, it suggests that maybe you could put people on the map with some decent accuracy. And so I wrote an algorithm that sort of tries to put people on a map rather than in that PCA space. And what you get is, this is, so say for this individual here, this is a Spanish individual. What I did is I pretended I didn't know he was from Spain, okay? And then I took this algorithm that I wrote, and I said, okay, algorithm, show me where you'd put him. Okay, pretending you didn't know. And it put him there. And then I said, okay, well now let me see what the label was. Oh, it's Spanish. Oh, that's not bad. It's on the Iberian Peninsula. And I did that for each and every individual. Sort of hide the information and then try to guess where they come from. And these are the locations of all my guesses, okay? And you do pretty well in that you get sort of for 90% of the individuals within 630 kilometers, okay, of the center of their country. For 50%, you're getting within 240 kilometers, okay? And it's not perfect, and it's not even that sophisticated. You can imagine I could do a lot better by just moving these individuals that I put in the ocean onto land, okay? So it's a very crude first pass algorithm, but it makes the point that you can, learn about the origins of an individual at a geographic scale that's much finer than just the continental scale. But there's a huge problem with that, which is with PCA, this particular method, mixed ancestry, leads to an intermediate positioning. So if I have an individual who's half Swedish, half Italian, I'm going to end up putting them right in between those two locations, okay? So I'm going to get it completely wrong. And as an example of that, and again highlighting how this stuff actually happens with some of the methods that are out there, this is someone I know shared this with me. This is somebody who has ancestry that's both East Asian and European. And they put themselves into this PCA-based method that is available online at 23andMe. So they took their genetic profile and asked, where do I sit in the world scale of diversity? Okay, so this is PCA results for the whole world. And actually, these are the HGDP populations again. And this individual, who's half East Asian, half European, pops out here in these brown populations, which are Central South Asian. So, if she went to 23andMe saying, what's my ancestry? And then she learns, oh, I'm Central South Asian. Gosh, that really messes with my head because I could have sworn my dad was, from Europe and my mother was from East Asia. So there's a lot of potential confusion in these methods. Now, to be fair, in the frequently asked questions at the bottom, there's a little kind of text addressing this issue of how you should be careful in interpreting this kind of map if you're admixed, okay? Have a recent mixed ancestry. And then just to highlight that there is a difference, though, between, even though PCA screws it up, you can tell the difference between an individual who's admixed and an individual who's somewhere along the gradient of the decline. That was your question before, right? And actually, you might be able to piece it together from what I've told you so far. Actually, does anybody want to take a guess? The haplotypes, right? So what haplotypes would you see if somebody was mixed versus they were from the middle?
You'd see one allele of each kind if you're admixed, or two of one kind if you're purebred, I guess.
Yeah, that's great. Yeah, not sure I like that word, but that's exactly it. So in our example of a Swedish and Italian, that individual would have a sort of, on one chromosome, have a string of alleles that are more Northern European, and then on the other chromosome, ones that are more Southern European. But if they were from, say, somewhere in the middle, like Austria, they would have alleles that are from Central Europe. Okay, that's great. So, just to summarize, so there's immense potential for personal ancestry inference, but it has numerous challenges. So obtaining the appropriate reference samples so that you're you're comparing any individual's diversity to the right set of global diversity. And really, a lot of it's about communicating to customers and about the education of science and to the public. And so what do we really mean by ancestry? How do we communicate statistical uncertainty? The particular limitations of any given analysis. Some methods have more issues than others. And then also, all of this can lead towards customers forming notions of group membership or race that might not, that don't reflect the reality. When they see those plots, they see these nice differentiated groups, and they're likely to forget how much of that variation is shared. So, okay, so another question. This one, I know you can't get right because there's not a right answer. So, what's your opinion? Personal ancestry inference is A, a waste of money, B, a harmless hobby if that's what you're into, C, fascinating, sign me up, or D, the first steps towards genetic elitism? Kind of just curious. Okay, so. You guys are getting fast. Okay. Oh, well, you can adjust them as you. Oh, okay. You see the influence of peer pressure, I guess. Okay, great. Cool, that's interesting.
So who said B? Jessica? You got about maybe 10 minutes, John.
Yeah, it's a harmless hobby because people are just trying to find out about their past. It's not really going to influence their own identity or the way they define themselves. So if they want to do it, be my guest, I guess.
What did you say, Faria?
I said A.
Oh, Dora. Why did you say? Oh, you said A, a waste of money. What about Faria?
I just stepped out for a second, so I'm sorry I missed the last few minutes.
Okay, what did
sorry.
Ryan say?
I think it's interesting. So I basically said something between B and C.
How about Dr. Harada?
Dr. Rada had an E.
Has E. Exactly. None of the above.
Okay.
We have about maybe 15 minutes at the most.
Okay.
10, 15 minutes. It's great.
Oh, okay. I'm not even going to bother with that one. Okay, so let me. Oh, this is okay. This hurts because it's like before dinner to talk about salivary amylase. I should have thought about that. Okay, but really quickly then, we'll sort of wrap up by talking a little bit about some of the interesting things about moving beyond ancestry, about learning about human evolutionary history from all this genetic variation and some interesting stories that come out. So in particular, another kind of variation besides microsatellites and SNPs that you probably haven't talked about is copy number variation. It's like microsatellites. There's a special case of copy number variation, but usually when it's being referred to in the literature now, it means large sections of the genome. And in particular, these are fluorescent in situ hybridizations, FISH results for the amylase 1 gene on different chromosomes. So on this individual, they have one chromosome with 10 copies of the gene, another chromosome with 4 copies of the gene. Okay, that's really kind of stunning, right? That you have whole copies of genes, not just little microsatellites of a few base pairs, whole copies of genes where your mother could have given you 10 copies and your father could have given you 4. That's really a lot of variation too in how much of that gene number you're getting. Then here's an individual with 3 on one chromosome, 3 on the other, so they have 6 total. Chimpanzees have 1 copy on each chromosome, so they just have, this is kind of your standard picture of a gene. You just have 1 copy of it, right? Albert.
What does this gene do?
Okay, so amylase is a gene that It's great, thank you. It is a gene that helps you digest starch. So and it's mainly expressed in your saliva. So that if you've ever done the game with the saltines, where it starts to taste like sugar, it starts out salty and then becomes sweet. That's amylase breaking down starch into sugar. Okay, and so. So this copy number variation, a big surprise of human genetic variation in the last 5 years is that copy number variation there's actually lots of it in the human genome, more than we ever had guessed. Most people have been focusing on SNPs, thinking that's the main kind of variation in the human genome, but actually there's lots of this, and now we have high throughput technologies for looking at it. And one of the interesting stories is this amylase one. So does anybody have any guesses as to what amylase might look like across the world in this copy number variation? Do you think it might respond to selection? It might. And so these groups went and surveyed amylase copy number in different populations around the world. And they looked at populations with high starch diets and low starch diets from very different groups around the world. So the high starch diets are like the Japanese eating rice, European Americans having lots of cereals, Hadza eat a lot of tubers. They're hunter-gatherers, but they eat lots of these underground starchy tubers. And then you have various groups that do not eat a lot of starch, like the Biaka and Mabudi, which are different pygmy groups. The Datog, which I think are pastoralists, they mainly have like goat herds and that sort of thing. And then the Yakut, who most of their diet is fish. So they don't have much starch, they're mainly eating proteins. And the average copy number of amylase is higher in all the populations that have a high starch diet versus ones that have a low starch diet. So it's a kind of neat example of selection due to diet leading to genetic differentiation at particular loci in the human genome. And then, let's see. If we look, and now if we just switch back to SNPs, okay, what that pointed out to us is that differing selective pressures around the globe can lead to differing patterns of genetic diversity that is going to be layered on top of the patterns that are due to demography and how we spread out across the globe. And so if you actually go through all of the SNPs and ask which ones are the most differentiated, and then ask what category they fall into. Okay, so we could say SNPs are non-genic, meaning they're nowhere near a gene. Genic, meaning they're very close to a gene. Intronic, meaning they're actually in an intron of a gene. 3' UTR, meaning it's just downstream of a gene. 5' UTR being just upstream. Synonymous, meaning it's in the coding sequence for a gene, but it doesn't actually affect the amino acid sequence. Did you guys go through this sort of thing? Yeah. And then non-synonymous, meaning it actually changes an amino acid. And what you find, if this is based on the HapMap populations, is that of the SNPs that have FSTs greater than 0.65, okay, have very large differences between populations, there's an enrichment of ones that are near genes and especially non-synonymous relative to the background, okay? And this is a sign, this is what you would expect if what's driving the patterns of the most extremely differentiated SNPs is not the background patterns of demography, which should affect every genic class at the same level, but selection, which is going to act on genes more than non-genic regions. Does that make sense? Okay. Demography affects the genome as a whole. Selection targets specific areas, and it's going to target genic regions in particular, and perhaps drive them to have higher levels of differentiation. And so one way then of finding genes that are under selection is to actually look at what genes are the most different between human groups, okay? And what you see right at the top of the list that pops up usually are a bunch of genes that are related to morphological traits, like skin pigmentation and hair development. You see immune response, DNA repair and replication, sensory functions like olfaction, eye development, insulin regulation, metabolic syndrome. All of these things are sort of believable that they've been under selection in different ways around the globe, right? We know that skin pigmentation is related to how well you can metabolize certain vitamins and responding to different levels of solar radiation. Immune response, there are different pathogens that exist in different parts of the world, and so might lead to selection that differs among populations, so on and so forth. And it's kind of interesting that what we see as our surface indicators of like how each of us is, when we look at somebody, are these things that are some of the most differentiated genes in the genome and are also have been under selection rather than just reflecting kind of the natural background variation. And so in particular, this is kind of the same point I made earlier, but this is a different way. This is using a particular browser of the HGDP, this Human Genome Diversity Panel I keep talking about, of their genetic data, where you can go in and look at any gene region and see these are all the genes, these are all the SNPs, and you can click on any SNP and get a map of its diversity. And you can see what the FST values are like across the genome. Okay? The scale here is kind of tricky, but just understand that it's been rescaled, so these are relative values. And you see how across most of the genome, it's really quite small. And so you pick a random SNP, and you get this kind of pattern, where for this one, there's a G allele and an A allele, and the G allele and the A allele are kind of found all over the world, although I guess the A allele is missing in a few populations in the Americas and a few places there, but it's also missing in a few populations in Italy as well. Okay.
Anyone can access this?
Anyone can access this, yeah. So I can give you the URL later. I guess I should have put a URL on there. So it's pretty fascinating because you can go through and look at all these different gene regions and see what's going on. Okay, so this is the ACA2 gene. And if you look here, okay, this is what most of the genome looks like. Or in fact, it even looks somewhat lower, right, of very low values of FST. Around the ACA2 region, they're slightly elevated. And then look at it. Here's a real zinger, okay? This is a real extreme FST SNP. And you look at it, and oh my gosh, this shows like some really strong differentiation between out of Africa. It's still not perfect, okay, in the sense of like, if we think about races and we want to say, okay, sub-Saharan Africans are a race and everybody else is part of some other race, it's found in other, the G and A allele are polymorphic outside of Africa, and the A allele is found within Africa, okay? But this is one of the most differentiated SNPs in the whole human genome. That's kind of as clear-cut as it gets, okay? And what does this SNP do? Does anybody remember the ACA2 gene from a previous lecture? Michael? Eye color, and also skin pigmentation, okay? So this part of the gene has a SNP that's involved in skin pigmentation, okay? So it's not surprising then that skin pigmentation, we know by eye that the genes controlling skin pigmentation must somehow be differentiated between African and out of African populations, right? And, okay, and, right, so ACA2 came up in a previous lecture for eye color and skin color. And then actually there's a separate, this is kind of, also gets to the complexity of genetics. There's a separate SNP in the same gene that affects eye color. Okay, it's not, and they're not necessarily perfectly tied together. And so this is one of the SNPs that affects eye color, and it's blue eye color versus brown. And so you can kind of see how that one's more common in Western Eurasia than Eastern Eurasia, the rest of the world. Okay, so, and this is, these are kind of these examples where, again, the most differentiated SNPs are the ones that affect morphology, and even in the best cases, the most differentiated SNPs are not really, they don't fit a sort of any ideas of racial categories. So just to conclude, I'm just going to jump down to the last point, which is intelligent views about our common genetic heritage and diversity are going to be crucial in this post-genomic world, right? We're really going to have it so all of our uncles and all of our relatives who aren't lucky enough to be in this classroom can kind of get the complexities of this information. So hopefully you guys can sort of take what you've learned in this class and help educate the people around you, because I think if there's a lot of misinformation, there could be some really negative consequences. So, okay, that's it. Thanks, everybody.
God, that was really terrific. I mean, it reinforced everything that we talked about in lecture and in class. Are there any last questions from Davis or here down in Los Angeles regarding this? I think that what John said in the last is, from this point of view of this class, of genetic engineering in medicine, agriculture, and the law, and the relationship between these issues in society, what John said last, and that last point right there, is absolutely one of the most important things that you have to take home from here, because one could get a very simplistic impression and make very wrong conclusions, particularly from, you know, very small numbers of genes or alleles that are highly differentiated, as we've been discussing. And so I think it's going to take a lot of information being transmitted, as John said, from here, as I said at the beginning of the quarter, to your mothers and fathers and sisters and brothers and aunts and uncles and cousins and friends and everybody else, in order to have a really intelligent view of human beings as we learn more and more and more and more about our heritage and origins. And John's really in the thick of all of this and making, as you can see today, some really pioneering contributions. We have a reception for Professor November outside with a lot more food this time than we've had in recent weeks, because it's been disappearing very rapidly. Ryan, Dora, John, thank you so much for listening. I hope you enjoyed this.
Oh, it's a terrific lecture. Thank you, John.
Thank you.
Thank you, guys.